Well, thank you very much for those very kind and gracious words, and I'm a very lucky person to be here, to have a second life, which will be quite enough for me to be here at BSMS until the next phase, whatever that is. We don't mention the R word here. So yes, and thank you also for making this possible. I thought when I did the first inaugural lecture I'd never have to do another one, but it's very different. And actually, it's been more fun because, of course, so much more has happened in this last 20 years than when I started. And I'll touch on this, the huge change that has come about in understanding. One of the themes will be, how has that change happened? What does it mean, and how do we cope with it? Because, of course, we all rather naively assume that change is going to be just simply more understanding and therefore more clout to deliver care for our patients, sort of treatments and radical treatments, cures. But actually, it's not quite as simple as that. And what we're having to learn to do is to deal with and think about complexity in new ways that we haven't really considered at all. And I think that's the challenge for many of you. Thank you, everybody, for coming. As a great non-attender of meetings and things, I feel ashamed or humbled to see that there's so many here, and I will do better in the future, I promise. The Dean made it quite clear there was no escape from this, but I did ask for extenuating circumstances, because most professors are of more tender years, shall we say. But no, he was inexorable. Well, again, just as a sort of preamble, it's lovely to see you all, and I know people have come from many different walks of life, and we've got people who I've met some years ago in a medical context, and they've come up here, and it's particularly nice to welcome them. It's lovely to see some old friends here, and we'll touch upon that as I go back. I am going to reminisce a little bit. And the point about reminiscing is, of course, as Karl Marx would have said, it's okay to theorize, what are you going to do about it? I paraphrase him, I think. There are experts in Marxism here who will correct me. I won't name them. So, neurodegeneration, it's a term which means what it says, that this is a process or processes whereby nerve cells, which you need for your day-to-day life, we assume, pack up working normally, degenerate, go through a series of changes and finally die. Now, it's very interesting to think about that. How do we know what happens once the trigger is thrown on neurodegeneration? We cannot see into the brain as it happens, or can we? That's again another theme. We can only we can only reconstruct from a series of snapshots what's happening, and that is one of the limitations of our work. We need to start, I think we all start from a foundation. So this is why I'd start with neuromythology, because much of what we accept will have to be modified. Some of it is pure myth, of course, which we generate ourselves just to impress, or just because we want to alter history. But some of it is, of course, real gold and needs to be carried forward into our current thinking. So sorting that out is important. I'm going to start with some past legends, and I'm sure many of you will recognize this picture of a morning clinical demonstration. So little has changed these days. At the Pitié-Salpêtrière, from which my friend Gilbert Ben-Simon has come over, a long-time collaborator, with Jean-Martin Charcot here, who was really, I think, arguably the founder of modern neurology. Many others contributed, of course, but I think he has a really star role. And he first described, in essence, motor neuron disease, although Martin Turner, who's here, has unearthed a very fascinating study to say that it was, and Michael Swash, a British person, after all, that did it. Gilbert, we'll talk about this afterwards. Lockhart Clark. Be that as it may, here is Charcot, and here are a couple of other figures in the history of motor neuron disease. Let me just pause there and say I'm going to talk about motor neuron disease, MND, and ALS, amyotrophic lateral sclerosis, interchangeably. So please don't think there's any subtlety in this. This is simply terminology, and we use the terms interchangeably, and I'm going to use them like that. So we can touch upon that if we need to, but I think let's just use those interchangeably. So Geoffroy, who was one of the co-authors on the original 1869 paper in which Charcot delineated ALS, and he wrote subsequent papers even with more studies. Pierre-Marie did important studies on motor neuron disease, ALS. Of course, the French called it ALS. It was the British who termed it motor neuron disease. And there are other names here, such as Babinski. I wonder how many people know what the Babinski sign is. This is really symbolizes the arcane nature of neurology. I see the dean laughing there. So what is the Babinski sign? Is when you get a patient half naked or totally naked sometimes, in the old neurological ways, not in these days, on the bed, and you scratch your Bentley key up the outside of their foot. And if the toe goes down, you sing hallelujah, shake the patient's hand, and off they go. If the patient's toe goes up, I don't recommend trying this at home, then it's the box of Kleenex tissues. This is bad news. And if, in the old days, of course, the neurologist would then say to the surgeon, and we have one, at least one here, mark a cross on the patient's skull, operate here, please. And this was the clever lesion. Of course, sadly, with the coming of MRI, we now know that we're wrong in most of these occasions, but it's still quite entertaining to try that. So that's Babinski. And our surgeons are very good at putting up with this and playing along with us. And there is Charcot with his halo. He is the hero of modern neurologists. And this is a paper by Martin Turner and Michael Swash, both of whom are here, in Brain recently, just to show that history sometimes covers up the efforts of very shrewd observers. And one of our duties, I think, is to unearth this hidden history and to pay justice to these people. And we've all come across people, usually at the Academy, American Academy of Neurology, who give a paper claiming to have described something which one's research fellow has described 2 years ago and written up and so on. I mentioned the one's research fellow because it's never the boss. Let's go on. That's Charcot again in his nice livery of the Légion d'honneur. So what was the nature of the condition which he delineated, motor neuron disease or ALS? The core concept, because it's that on which we have to build to change our concepts and move forward. Is it the right concept or does it need changing? And the answer is, you can guess, it needs changing. So what Charcot described, this is the spinal cord in cross-section from a human being, or a human who's died of motor neuron disease, and the shadowing there is what was called sclerosis or hardening of the lateral columns. And the amyotrophy is due to degeneration here. This is the white matter, although it's grey in the picture. This is the grey matter in the spinal cord, which has the motor nerve cells in it. And I've blown it up here. This is a section of our own series of autopsy cases of MND. This is a relatively well-preserved spinal cord because these little guys are motor neurons here, and I've blown them up large here several hundreds of times to show you the nature of these lower motor neurons. So amyotrophic lateral sclerosis, if you lose these, you're going to waste and become weak. If you get damage to these lateral columns, that's sclerosis or hardening, degeneration there, you're going to get what we call spasticity, or the upper motor neuron syndrome. And this probably originates from this area of the brain. This is a cross-section of the brain here, the motor cortex, and this is one of the big motor neurons of the cortex here, one of the giant pyramidal cells in the motor cortex. And essentially what motor neuron disease, the core concept, is degeneration of these motor neurons. The tracts that pursue their course down through the brain into the spinal cord, their parent nerve cells are these ones. And these neurons, which are the lower motor neurons. And it was the paired degeneration of these systems which gave the characteristic clinical picture recognized by Charcot and by all of us. Now, for many years, although there were exceptions to this, this picture of the patient presenting with a combination of upper motor neuron features, typically stiffness, brisk reflexes, and so on, and wasting and weakness due to degeneration of these lower motor neurons, was the rubric, and the only rubric. There were whispers that it was more complex than that. There were people early on, including Alzheimer himself, who described Alzheimer's disease, that indeed some patients might become demented. And there's a very nice description of a patient with a frontal dementia by Alzheimer in the early part of the 20th century, which was put down to syphilis, because everybody thought, you know, everything was due to syphilis in those days. Probably it wasn't. And then subsequently a series of publications, but not hitting the mainstream literature. So we all said at least one thing about this condition is that your brain will remain intact, that you retain your marbles, if you like to put it bluntly. But it's a bit more complicated than that. So that's one thing that we've had to rethink. The second point really is that we have to consider now whether this is what we mean by a disease or a syndrome. Everybody, I think, is familiar with an icon in the public domain here, Stephen Hawking. But in America, it's Lou Gehrig, and it's often termed Lou Gehrig's disease, who is probably the major icon. He was a baseball player, really one of the best of all time, and he went down with motor neuron disease and died after a couple of years of illness. So everybody knows about that. Obviously, Stephen Hawking has a somewhat unusual form of the disease, if that's what he does have, but he says he has himself, so I think we have to accept that. Within the rubric, though, sometimes patients present with speech and swallowing problems, the so-called bulbar onset form, and this shows the wasting of the tongue of a patient who had the bulbar onset form, and they would have very brisk reflexes around the jaw and the face and so on. And they may or may not have weakness at that stage in the limbs, but sadly weakness would follow. This is the limb onset. Here you see just a hint of wasting here. This would be an early feature of motor neuron disease. And there are many other, before everybody panics, there are many other possible causes of weakness and wasting, so please don't, you know, go assume the worst. But then and again, right since the early days, this is a patient with weak legs, weak and wasted legs. And this had been recognized by Pierre Marie, who I referred to, back in, I think in 1916 was the publication. Although rather neglected. It was known as the polyneuritic form of ALS or MND. And then this, and we'd written about this in the 1990s with Michel Hu, the so-called flail arm, recognized by Gowers here. Gowers was a famous neurologist at the National Hospital, writing in the 1880s. This was our patient. But you see here, the patient can stand up, can hold his neck up, but his arms are profoundly wasted and weak. So the different, what we call phenotypes, clinical presentations, are important, and it's particularly important for the patients. The problem is with all survival studies, because we're going to come back to some of these, I want to explain this a little bit. So here you have what's called a Kaplan-Meier curve. So you have the proportion of surviving. So supposing 1,000 patients walked into your room on day one, and they were all the patients you were going to see for the next 5 or 10 years. They'd all be alive then, and then you would follow them up over this would be 10 years, 5 years here, and so on. And they would, unfortunately, some would survive and some wouldn't. And you see, what you're seeing is the falling off of survival. It starts at 100% when you see them, if you would see them all on that day, and it goes down to not quite 0% when you get up to about 10 years here. But some patients do better than others. So these patients, this is what we call the flail leg. I'll just get rid of that bit. Which you saw here, and then the flail arm, which you've got there. But if you've got this, the bulbar onset, you are much more likely to die at a given point earlier than if you've got these other forms. Now, there's considerable range there, but even when you're counseling patients, that is quite important to know. And it's also, it could be important biologically. So what is the reason? Is this just some random stochastic effect? I don't think we think it is. This was work done on our King's database with Ammar Al-Chalabi, who's now professor and head of the MMD clinic there, and Lokesh Vijayasakira, our research fellow, and a number of other members of our team. And I think that was the first time we had really robust ideas of survival. And this was a group of about over 1,200 patients. So it's pretty robust, I think. But what was particularly fascinating is when we used a statistical test, and I know there's a mathematician here in the audience, so I have to be careful, which I can't claim to fully understand. But essentially, this is a this is a complex statistical analysis called latent cluster analysis, in which you essentially feed the data on your patients in the things that influence their prognosis, their survival. And there are many: age of onset, duration of disease, etc. You can imagine an array. And you ask the computer to sort that out, the mathematical model, to sort the patients out into groups without knowing whether they are bulbar onset, limb onset, flail arm, and so on. You give the computer that task, and it does it like that. So you get groups according to the clusters, where you're clustering together the patients who are most widely separated, if you like, in these particular factors. And then we tested this, was Amar's work collaboratively with Lokesh and Dr. Ganaslingham. It actually, the machine phenotyping computer, this is a correlation with survival. Here you see the survival curves, does it better than eyeballing the patients. And my point there, I haven't got time to dwell on that, is that we do need alternative ways of thinking about what we see in front of us. It may not be as biologically significant as we think. And I think one of the ways that's going to feed forward into understanding the disease is to understand the biology of the phenotype in relationship to the genotype and indeed the epigenetics. And we'll come back to that briefly. So all I'm saying is that sometimes a machine approach which doesn't have the biases that we do as physicians, which we interpret what we see in front of us, may be helpful. We haven't proven that that is the case. I want to now just mention very briefly a couple of conditions which we've already also worked on, and I mentioned that in my abstract. One is multiple system atrophy. This is a gentleman whose neck has fallen forward. He's Parkinsonian. He's probably unsteady. This is what we call Parkinson plus syndrome, and you get Parkinsonism, unsteadiness or ataxia, autonomic failure. And in the brain, you get a particular molecular pathology, and here is the discoverer of this, Peter Lantos, who's professor of neuropathology at the Institute of Psychiatry, a couple of years ago. And these are the PAP-Lantos bodies. And these are blobs. And there's a lot of blobology going on in this. In cells. And these are not in nerve cells. These are in the supporting cells, the oligodendroglia. And that's an interesting variant of neurodegeneration, where the obvious pathology is not in the nerve cells which are degenerating, but in the supporting cells. Now, the nerve cells do indeed die, so how that happens we don't know. So that's, and this just shows a shrinking of the cerebellum in this so-called hot cross bun zone, which is one of the features you see on MRI. So, and the other condition was, in some ways similar, is progressive supranuclear palsy. These are all indigestible terms, I know. And this was described by John Steele in 1964 when he was a medical student, so there's an incentive for you. And unfortunately, Richardson and Olszewski are no longer with us, but John Steele very much is, and is an old friend. And this is a fascinating, there's John Steele. And this is a condition in which you get a failure of voluntary upgaze because of degeneration in the brain stem of a particular group of nerve cells, Parkinsonism, early falls, cognitive changes, and the molecular pathology, again, is very distinctive. It's a tauopathy. I mentioned the other one was an alpha-synucleinopathy, so a different protein gets tangled, coming back to our theme of tangles, and untangling them. So just leave it there, really. So, coming back to, I have to talk a little bit about my experiences, because it may be of use in helping others to avoid mistakes. I can't think there's any other purpose of doing it, because so the ideal career pathway for aspiring young clinical neuroscientists or neuroscientists is this. That's the Morris water maze. So you put a rat on a little platform, and it's not water, it's milk, so they can't see where they've got to go. They can't see the platform, they have to swim, and they have to find their way to the other platform, and they do it by trial and error. So of course, with the super smart young neurologist, for example, or general physician, there's the mentor, here's the young neurologist. I couldn't find one suitable, one of a female. I thought about looking up young Amazons, but I wouldn't recommend that as a Google search. So there's the mentor, myself, a sort of aged figure, and Telemachus starting off here, and they got to get to the chair in neurology there. And of course, along the way, they had prestigious awards, MRC Wellcome Trust Fellowships, no problem, and then they arrive at their senior lectureship and their chair. So that's the ideal one, and that's what you should all aspire to. I'm afraid mine is the alternative pathway, and well, it was fun, but it wouldn't be recommended. But in the end, it ends happily, because there we are. So there are various pathways into academia. So you can choose whichever you like. I'm not saying which is more fun. All I can say is I've had great fun doing it my way, so to speak. So now I want you to deconstruct this. This is an important figure in my evolution as a neurologist. And you might like to pause for a few moments and do a little bit of deconstructing, a bit of Sherlock Holmes work on this. This is perhaps unusual in an inaugural lecture. It's a picture of its era. I don't know if anybody would like to guess when that was. Anybody like to? If not, I'll tell you. 1960s, I hear. I think that's right. The building, perhaps 1930s. Yeah. Now, would you say, this is a doctor I'm telling you, so what specialty would you think would be likely? Here we are, pipe smoking, smart white coat. Psychiatrist, anyone? Not many. Neurosurgeon? Yeah. Neurologist? Well, there was a psychiatrist, actually. It's my father who's a psychiatrist at the Maudsley Hospital, and this is the Guy's Maudsley Neurosurgical Unit behind there, where neurology, when I went to the King's as professor, succeeding David Marsden, was housed in there. My father was consultant there, and he did have a great influence on me, because I always wanted to be a neurologist, and I presume I have him to blame for that. I also have him to blame for becoming an academic. Although he did considerable amount of research, and I'll touch very briefly on that, he was a very pragmatic man. He was a Mancunian, and he retained his Manchester accent to the very end. He had a very pragmatic approach to psychiatry, pragmatic meaning that if somebody wasn't well within a week, they would be told that they would be out on Denmark Hill promptly, and the registrars had to meet those targets in those days. But actually, he was a very kind man. As I discovered when I took over his patients, when I went to the Maudsley and King's, and there were about 3 or 4 of his patients who'd had chronic depression and anxiety, and they all came in to me and complained that I wasn't nearly as nice as my father. And I looked through the notes thinking, well, they must have had something done to them, psychosurgery at the very least, or ECT. No, they'd had the occasional touch of phenobarbital, the occasional little bit of Valium. Otherwise, it was the talking therapy. And this was not psychodynamics, I can tell you. This was Mancunian talking therapy. This was psychodynamics with a Manchester accent. But so he taught me to be kind to patients. I think the other thing was that he had good contacts. He'd been a neurologist at the London, not Royal London Hospital then, after going through the war as a neurologist in the Far East. And he'd met the dean, the future dean of the London Hospital Medical College, Jimmy Crawford. And so he took me along to see Jimmy and said, I brought my boy along. We were in the jungle together. Can you take him on? How refreshingly unbureaucratic, John. I think some A-levels were possibly mentioned, but that was a subsidiary, really. So he was at the London Hospital, and I was eventually. Catherine virtually demanded, my wife, that I should hand myself into the GMC, belatedly, but better late than never. Just to, again, this picture is of Thomas Willis. So although he was a psychiatrist, he was a neuropsychiatrist with a solid grounding in neurology. This is Thomas Willis. I now have a print of that myself, well, his print, and that's the Maudsley. And in the basement there was neuropathology, and that's important because he did neuropathology for a year or two before he became a psychiatrist. He was transitioning between neurology and psychiatry. And this is Leigh's disease. He described this disease, subacute necrotizing encephalopathy, and I was brought up with this disease, so to speak. There were always papers, reprints on the breakfast table with the Times. And this was a single author paper. His Professor Alfred Meyer, who was a refugee from Nazi Germany and a most kind, wonderful man, simply said, here's an interesting case, work it up. And it turned out to be a new disease. And it's now called Leigh's syndrome. There are now 20 or 30 different mutations, and it's one of the mitochondrial cytopathies. And indeed, nobody has ever seen these slides before because I photographed them myself. In his effects after his death, I found the slides, the original slides from 1950, from the child who died in 48, in fact. 47, sorry. And I photographed them, rephotographed them on my Zeiss color photographs, and so there you are, this is the first. And what I'm doing now is we're going to try and sequence try and find the mutation by getting DNA from these. If the police can do it from tiny samples, I think we can do it from sections in celluloid with Billy DiMauro, who's the leading expert on mitochondrial encephalopathies in Columbia University in New York. So just going on to the London, that was not the Royal then. There were extraordinary neurologists, one of whom I knew, that's Georgie Riddoch, or wee Georgie Riddoch, who apparently spoke the Doric dialect, but was said, once one could understand that, he proved to be an excellent teacher. So whatever that means. And there he is. There he is in the Bierstead Lecture Theatre, where I spent many happy, sleepy hours as a medical student giving a demonstration. And this is Lord Brain, who I met. And this is his last demonstration, it's said to be. And if that's the case, I was there myself. It's a scary thought to think that. Lord Brain was an extraordinary man and described essentially the paraneoplastic syndrome and much else. And was Michael Swash would have known him as well, I'm sure. At the London, though, although neurologists were pretty scary, they were a very kind lot. That's Chris Earle, Ronald Henson, I haven't found a photo of. But this man was our professor of pathology because after training at the London, it was accepted that after a year on the house, surgical medicine, you went and did pathology for a year. And this is where I met Michael Swash. Israel Doniak was an extraordinary man, one of the kindest and cleverest people I met. And I learned a trick from him, so that when you looked down a microscope with him and you saw something unintelligible down the other end and you said it was a colonic polyp and it was really a brain tumor, and he would just stroke his chin and he would say, nearly right. And so that is my formula now. So, you know, never say they're wrong, nearly right. A great man. And then is Professor Henry Urich, again, a very striking individual. He always seemed to me the apotheosis of a Polish aristocrat. He probably was, I think he was. But he was one of these neuropathologists who was very learned, but he slept through lunch and he would prop himself up on his microscope with his eyes resting on the microscope, so that if you looked through the door, you know, he was obviously, but if you pushed in, he would fall over, and so it was a bit. And he would sit at the back of the autopsy room, fast asleep, but bolt upright. But of course, if you asked him a question, his eyes were open. It was usually just to annoy the neurologist by telling them, yes, they'd have to wait 3 years for the histology on their brain of a particularly interesting patient. And that's Michael Swash and Caroline's wife. Michael was sitting next to me here, and we've known each other ever since. He swore I would grow up eventually, but he was wrong, as usual, about many things. We forgive him that. We've had many debates and agreements and disagreements, all amicable, and that's his wife's stained glass. Caroline is a very accomplished stained glass artist. I won't say anything more about Mike for fear of embarrassing him with riches, because he's been a great influence and indeed helped me in the research, and where I'll come back to. And so then it was on to the Hammersmith. And this was an extraordinary experience. You'll recognize the Hammersmith, many of you. This was where you went once you'd done, you know, certain jobs. And I applied for a medical job, but I got a job with this gentleman, Chris Pallis, Dr. Chris Pallis, who was quite an extraordinary teacher, a wonderful man. And I was very fond of him, but he wasn't an easy man. And you'll see why. So in 1968, this is Paris in 68, he disappeared for a couple of weeks without leave, and he was on the streets with the students. Just in case you wondered which side he was on. Being a neurologist, you might have wondered. But he was on the side of, well, whichever, the left. This was his nom de plume, Maurice Brinton. So in his office at the Hammersmith, he had two big filing cabinets. One was full of the most wonderful teaching slides on neurology, and he did wonderful sessions every Thursday afternoon. The other was in his tracts. and he belonged to an alpha-radical group called Solidarity. He flirted with me, I could say, in terms of seeing whether I was suitable material, but it soon proved that I didn't have an understanding of Marxist-Leninism, which was up to his, and so luckily I was passed by. But there were many interesting moments at the Hammersmith. He did write a book on brain death, and I think he influenced the whole field of diagnosis of brain death, although the way he looked at one often wondered at the end of a wood round whether he was wondering at what level of decerebration his SHO was functioning. Then there was Donald Carl, who you've mentioned, who indeed was a seminal influence, and he was a wonderful clinical researcher, and he did introduce the world to dopamine agonists. So you've heard about levodopa, but also the idea was to try and use drugs acting directly on dopamine receptors. And I was a very minor cog in his machine there, taking part in the trials. But we did introduce bromocriptine. It was my first Lancet paper and almost my last. Donald went on to do great things in the movement disorder field, and I haven't seen him recently, but he went on to become professor at Vancouver, and certainly was an example of somebody who thought very hard about experimental medicine, as I'd call it, experimental pharmacology. So yes, you're absolutely right, Michael. Now we step back to the things that didn't come out in the interview with the vice chancellors and with John just as well. So when we were this was the sort of early 70s, and at that time, as you'll all know, there was Southern Africa was in turmoil. It's amazing to think now that most of it was still white dominated. We were involved in groups which were supporting the liberation movements, Frelimo, the Front for the Liberation of Mozambique, MPLA, the popular movement for the liberation of Angola, and there was another movement for Guinea-Bissau. And there was a group in London called CUFMAG, the Committee for Freedom in Mozambique, Angola, and Guinea. And we were no doubt naive and so on, but it was interesting times, and we raised money and so on. But at the same time, we were also in touch with people who were living and working in Africa. And one of our colleagues, John Yudkin, who became professor of medicine at UCH and the Whittington, encouraged us to go. He'd already been to Tanzania. He'd done VSO there. And on his encouragement and talking to the dean of the University of Dar es Salaam, the medical school, who came around and recruiting, I remember we met at the Hotel President in Bloomsbury, and he sort of said, well, we'll sign you up. And we said, what about the conditions and terms? And he said, oh, well, don't worry about that. Well, we never saw those. I mean, that was it. But somehow we got paid, and was supported by the English universities scheme through the Department of Overseas Development. And off we went. As I said, Tanzania it was then a source of great debate. Nyerere, the president, was promoting African socialism, Ujamaa, or family socialism, loosely translated. That's Samora Machel from Frelimo and Agostinho Neto from MPLA. That's the University of Dar es Salaam. I think that's Hekima ni Uhuru, which is knowledge is freedom in Swahili. It's a beautiful campus. But as I said, you know, there were difficult times, and these countries had active liberation struggles and much bloodshed. And of course, you know what was happening in South Africa at that time. So we came to meet with many of the people involved in the struggles, because the liberation movements were many of them were based in Tanzania. We met a lot of friends who have remained lifelong friends. Well, I haven't seen Wen Kalama for long. He was sharing the same floor on the flats, and we looked after each other's children. He's professor of parasitology and is now was trustee until recently of a major malaria charity after he'd retired from the university. A marvelous man. John Yudkin here, who influenced us. Walt Willett, who's at Harvard, is one of the most highly cited professors of nutrition, and of course he was just one of our mates then. We didn't know that he would go on to such eminence. Julie Cliff, who's professor of public health and epidemiology at Maputo, spent the rest of her life in Maputo, although she has links with Melbourne. And a particular friend, can't be here, Henry Bernstein, who's professor of development studies at SOAS. And I went to his inaugural, which was about the peasantry. And it was very interesting, but I didn't understand much of it. It was mainly a theory, economic theory, but it's a fascinating field. He did have a lord, I'm not being critical, he did have a lord attending his inaugural, but the lord was fast asleep, sound asleep throughout it. I won't give any names. So just to show you that there are constantly, sorry about this. So this was the out of office activities. Embarrassing. Camping in the game parks with all the latest modern equipment for camping, including a Renault 4, which persistently failed on us, but never quite got, we never had an elephant actually catch up with us, although they tried. Camping, and this was Irene, my then wife, and our little son. This was about 30 seconds before a troop of about 15 wild dogs came rushing through the camp. Luckily they were after an impala rather than Pierre's, and he just stood there watching them as they passed. On the beach in Zanzibar with colleagues who were, this was a postgraduate fellow, a postdoc, and then I think this is the sundowner walk along the beach at Zanzibar. So if you were doubting about going out, there are good reasons to be out there, and Tanzania and East Africa is extraordinarily beautiful. We also met some very impressive people. I can't say they were close friends, but we spent some time. One was Ruth First. Some of you will know that she was a leading academic and writer, journalist. She was imprisoned by the South African apartheid regime for 117 days in solitary confinement, and wrote a very eloquent book about that. And she was a major activist for the African National Congress and the anti-apartheid movement. And there's a very touching film, a very good film, I think, about her experiences and those of her husband. So we got to know her a little bit, and back in London again. The curious thing is, this is what happened in Dar es Salaam. We just had our second child down at Ocean Road Hospital down the road from us. No blood, no transfusion. My Irene had quite a severe postpartum hemorrhage, saved by a very excellent young SHO who managed to sort everything out. Eternally grateful to him. And so we came back in our little Renault 4, and about 4 hours later there was a knock on the door, and in came Ruth First with another colleague who used to be at Sussex, Professor Malcolm Siegel. He was in development studies, very eminent, and they sat down and proceeded to have a cup of coffee with us, despite the fact that my wife was then looking extremely pale, and we had no food in the house, and they wanted to debate the disagreements between Trotsky and Lenin, as one does. Unfortunately, we were unable to join in this conversation, and we could only find eggs and bread, so they had boiled eggs and dipped soldiers, and went on with their discussion. But it illustrates, I think, the sort of place it was. Joe Slovo, we knew very slightly, others here knew better. He was head of the arm wing of the African National Congress, and he spent a lot of time in London and in safe houses. Ruth First was assassinated by the South Africans in a letter bomb, and he was constantly on the alert to this. We did get involved through the ANC with a charity called Heart, which was set up just after the release of Nelson Mandela to raise funds for health and education in South Africa. And I worked closely with this person I haven't seen for some years. Her name is Nkosazana Dlamini Zuma, who is now just been elected president of the African Union. And she was a tremendous person. She became Minister of Health, not during the AIDS crisis when there was the denial of AIDS, of HIV virus causing AIDS, but before that she was always very good, and I think she's a very impressive person. So that's another contact we had. And then subsequently I worked at Mulago and did teaching there in Uganda. This is the big teaching hospital in Uganda. My wife also spent a year there, and there we are on the wards with Jack Frears, one of the other teachers, and the medical students. And it reminded me how much I enjoyed teaching, and I perhaps ought to get back to it. But that's a great experience. Coming back, I want to romp through this. It was very difficult to get back into neurology, not being known at Queen Square, so to speak. But one wonderful person, this is Atkinson Morley's Hospital, which was the neurological wing of St. George's, and that's Godfrey Hounsfield, who actually designed and put into practice the first MRI scan, and that's the first clinical MRI ever done with a hole here. That's a tumor, I think. So that was going on, and then this lady, Pauline Munro, was a wonderful influence. She simply took me on board, despite the fact I'd come from nowhere, and said that I could be the locum senior registrar, and then pointed me to the substantive post. She also gave me the first intuition about how one might look after patients in a multidisciplinary fashion, and that was absolutely ingrained in her practice. Very interesting person. I haven't got time to go on about Pauline, although I'd love to. She's still active. She has a charity in Russia. She has a flat in St. Petersburg, and she's still promoting multidisciplinary care in Russia at the moment. I spoke to her last night. So she said, well, you ought to go and do a PhD, so the best person to work with is David Marsden. And you can see whether or not he really danced the samba with Dolly Parton in Rio only 4 hours before his keynote morning lecture on basal ganglia disease is unimportant. The fact is that David Marsden's colleagues believed it was well within the limits of his possibility, and it certainly was. I suspect he did, because I've seen him dancing at other meetings up to an hour before the lecture was due to be given. There's many stories, but I won't give. David was undoubtedly the leading figure in neurology of his time in the UK and arguably worldwide. A wonderful clinician, a great scientist, and Hans Gobber, who's here, worked at roughly the same time. I was given the circling rat model of basal ganglia disease as my project. Well, yes, you can imagine putting that to neurosurgeons and neurologists at an interview. Yes, I worked on the circling rat. There was a reason. Essentially, we were trying to work out the outputs of the basal ganglia, which is the bit that goes wrong in Parkinson's disease, because we knew that there must be pathways going down. So that's substantia nigra. That's the bit that dies in Parkinson's disease with the dopamine fibers. And then the outputs go through the thalamus and back up to the cortex, but not to the motor cortex. So we knew that there must be pathways going down to the brainstem, which are very important in motor behavior, and that's what we set out to do, and I won't go into that further. We made a number of conclusions, and luckily I wrote some papers, because I remember Michael Swash telling me it was high time I wrote some papers about them, and he was dead right. Then I went, again, seizing opportunities. We were all frightened that we'd never get a consultant job, and I think the same applies to trainees now. It probably wasn't true, but you didn't know it at the time. And I went to Southampton, where this was a real induction of labour, because it was a very busy practice, and I'm glad I went for 4 years as an NHS consultant, subsequently senior lecturer. But then Pauline said we have a chance to come back to St. George's as senior lecturer, and this is really where I think my science, such as it is, got going. And this is Brian Anderton, who was the professor of neuroscience, and he introduced me to tangles. And these are tangles in Alzheimer's disease, in cortex there. You can see the dark blobs there, and more blobs here in Parkinson's. And one day he said, come and have coffee. And he was saying, well, why don't we do something on motor neuron disease? And I said, well, there's nothing much going on there in motor neuron disease. There's no known molecular pathology. And he said, oh, well, there might be. We ought to look at some of our antibodies. He was making monoclonal antibodies to all these things. So I sort of said, I'll go away and read it. And of course, straightly I did. I realized he was dead right. And the question was, what was in these? Were there molecular signals in there? And the next stage was to use an antibody which he just made against this protein called ubiquitin, which basically sticks to tangled and abnormal proteins. So it's a good marker for something going wrong in a cell. So we had this antibody, and I remember a Saturday morning in Tooting, in the basement of the medical school, using an old Zeiss from Professor Nigel, the professor of anatomy, which he didn't seem to use. And I looked down and I realized that this was something that nobody had seen before. And you may not think it's very much, it's just a tangle or a blob, but to me it was a eureka moment. And there aren't many anyway in my life. So it may be a little sad, but it was the turning point, because it turned out that these were the molecular hallmark of motor neuron disease. To cut a long story short, we didn't do the experiment that John Trojanowski did 20 years later. carry on making monoclonals against these purified things from the frontal lobes of patients with a related condition, frontotemporal dementia, isolating the monoclonal antibodies and finding out what they were directed against. And they found that the major protein component of these, apart from ubiquitin, was TDP43. Just bear that in mind. I'm going to just skip over these quite quickly, but just to mention some other heroes in my photo album. This is Bud Rowland, still going strong at Columbia, New York, Columbia Presbyterian, the New York Neurological Institute. And here he is with some other luminaries of neurology. And the reason he was so important is that when I just started on this in the early 1988, when we discovered the ubiquitin story, I was sent over to a meeting and he was there. And he was really a very, very senior figure in neurology. But open arms, with an open arms policy for anyone with anything to say. And he's been an immense support ever since, and will be to many others, including Martin and other research fellows. This is a guy called Forbes Norris, and this is the Pacific Medical Center in San Francisco. And he was really the originator of multidisciplinary care, a very sophisticated form in the States, and he died in 1993, a great man. And this is another great hero, Asao Hirano. This is his Hirano body here. These are another blob. These contain actin. He's a most lovely man, and here he is. And I show this not to show me, but to show this is his tie with the Hirano body sewn into it. Wonderful idea. I haven't yet got one with my skeins, that we call them, the inclusions, but I think we ought to do that. So Asao Hirano told me this is the island of Guam, as you may recognize from the air. So Asao Hirano was, in 1960, he was told by his professor at Montefiore Hospital, New York, to go off to Guam and solve this curious disease which was killing people with Parkinsonism, dementia, and ALS, all together. Fatal 3 disorders, flesh there, all contended, to quote Gerard Manley Hopkins. He didn't tell his wife, he'd just married, that he was going there to work for several years. She thought she was on honeymoon until, no, I'm joking, not, until she found he was doing postmortems at all times of the day and night. He also told the story, this was last year, a wonderful story of how there wasn't very much to eat. So he sent one of the technicians, had a gun, and he sent them into the jungle. And instead of coming back with a fruit bat or something to eat, he came back with a Japanese soldier, alive. And then he said, well, 2 weeks later, he sent him out again. He came back with another one. So there were 2 of them, and this is true. These were soldiers who had been surviving in the jungles of Guam for 15 years. One of them went on to become a very famous figure, a senator apparently in Japan. This is the disease, and I'm not going to say anything more. This is a devastating disease, but one of the extraordinary puzzles of this field is that this is just ALS, because you could get ALS, you could get the Parkinsonism and dementia, and you could get them separate. I won't go into it in detail. And this is over the years, 1945, and this disease had been known since the middle of the 19th century from records of people passing through. It's virtually disappeared. We still don't know why. Don't know whether it's genetic, whether it's... This is John Steele, who we saw earlier, who described Steele Richardson, and he now lives and works on Guam, has done for the rest of his life, and has done much of this research. Curious. Right. So back to the Maudsley. I was very lucky to succeed David Marsden. A difficult act to follow, and one I wouldn't even try to justify. But we did eventually build up a unit, which Michael, well, it was very kind in describing. It was more modest in my view, but I wouldn't gainsay him. These were some of the people we worked with. That's Catherine, who's one of our research fellows, and Martin Turner, who's here, and John Q, who very tragically died of melanoma a couple of years after working for. He was my first MRC-funded research fellow, and a marvelous man. He did make the big mistake once of getting upgraded on a flight to Los Angeles when I wasn't, but I forgave him that because his other attributes were so positive. Peter Anderson is a wonderful geneticist who we work with, and I'll very briefly, I'm going to scoot over some of these just for time, but just show you, just give you a soup sum. So this is a PET scan, and the bright areas here represent binding of this thing, which is a GABA active drug, and this is binding to, we assume, areas where there is a deficiency. This is a decrease in sporadic MND. But and this was work of Martin Turner, although Catherine had done earlier work using the same PET ligand. And this was looking at this molecular variant. This is a SOD1 gene mutation of a peculiar type found in northern Sweden. This is where Peter Anderson comes in. They're all brothers and sisters genetically. They all have this shared genetic type around the SOD gene and many other areas, and they had quite a different profile of binding of this particular ligand. And we even were able to show, although this, of course, very small, I think there was only one case there. Is that right, Martin? Two, sorry, thank you. Where pre-symptomatically they had some change. Now, that's where the future lies, and I think people like Martin will be pursuing that with big international cooperative studies to try and get at the pre-symptomatic changes that we have. And I'll gloss over these, Martin, I'm sorry to do it just for time. The other important thing was that we got our psychologists involved, Laura Goldstein, who's now a professor at the Institute of Psychiatry, and Sharon Abrahams, who's a senior lecturer in Edinburgh, and she was able to devise this neat little verbal fluency index, which is not a shortcut for detailed cognitive studies. We did those as well, but it enabled us to dichotomize patients into two groups, either impaired or not impaired, and to do studies. I'll just show you an example. And then we had imaging colleagues, Steve Williams, Derek Jones, and Andy Simmons, and this was the new unit that they built through their own efforts. And this shows one of the techniques called diffusion tensor imaging, whereby you can not only measure the molecular changes, if you like, in these pathways. This is the corticospinal tracts, but actually draw them out. And this is a technique which I haven't got time to go into, but which Mara Sercignani here is expert in, and which we will pursue. The first work done with this was by Kathy Ellis, who's a research fellow, who's very good at eating cherries and even better at doing MRIs. Here we are eating with the family. And she showed, the first paper of its kind showed that there was a decrease in this particular measure, which looks at the integrity of the pathways that you want to look at in ALS compared to controls. But what was very interesting is that these patients with this particular mutation were much more like controls. And we can speculate about that, but it may mean there's a different type of pathology going on in these different groups. One dying back, one perhaps dying forward. I haven't got time to go into that. I'm going to pass over these very briefly. I'll just show you a picture, because I like it, of Paul Wicks, who is one of our postdocs who went on to great things, as you can imagine. Satomi, who is here, did some very nice work counting neurons. This is not something one wants to ask people to do unless you trust their good nature. And we do. It takes a lot of time and effort. But she did some very nice studies published in Brain. And what the key point here is that not only was there a decrease in the area 4, the motor area of the cells, these are these big cells here in motor neuron disease, but also in area 9, which is in the prefrontal region, the prefrontal cortex. In other words, MND is not just a motor disease. And there's much more of that. I'm just going to wind up really with saying one or two words about other mentors. Gilbert Ben-Simon, who's here, from Pitié-Salpêtrière, Vincent Manager from originally Hôtel Dieu, and for many years at Pitié in Paris. And this was a seminal experiment they did, which was a trial, the first modern robust trial, I think, of Riluzole, which is now licensed medication. It's not great, it doesn't save lives, but it does do something. And this is their study in the New England Journal, and showing that here we have the survival in the treated patients being better, and the controls here in a very small group of patients. And this set the cat amongst the pigeons, and we went on to do a bigger study, and they included me very kindly. This is actually a meta-analysis. So here we have the survival again, and this is the active drug, and this is the placebo, and that is now treatment. I'm going to gloss over this again just for time, but just to say we went on to study MSA and PSP, which I talked about before, because they were good targets. We could measure progression by survival. Everybody told us we couldn't do it, but I'll go straight on to the design. We did. The primary outcome was survival over 3 years. But we also measured a lot of things. So we randomized patients with Parkinson's plus, stratified them, and then followed them for 3 years. This was a big study, 44 centers. It was something of a nightmare to coordinate, but it did work. And at the end of the day, I will bypass that, to our surprise, the clinicians were pretty confident in the diagnosis. So what this shows is the diagnostic probability here. So you had to classify where you were along these visual analog scales. And this is the PSP and this is the MSA. But when they appear to you, they can be very difficult to differentiate. In fact, they got it right in about 90% of cases. And what's more important, so did pathology. And I think we have about 150 brains now, but when we published, it was 112, and there was 90% accuracy on the criteria we had. Sadly, this is survival curves again. There is no difference in this case for riluzole between placebo and riluzole. PSP, 362 cases, MSA, 398. This is by far the biggest series. And we've done a lot of other things on it, genetics and so on, because this is a unique cohort. And one of the things for the future is to try and create these cohorts. And it isn't trivial, but it is doable. That's what it felt like, trying to get it published. We all know that feeling, I think, but we did. So the fruits of NIPS, well, they're ongoing, and we've published a number of things which I think will modestly change the scene. New diagnostic criteria, new functional rating scale, properly validated, and genetics, which is ongoing with Amar Al-Chalabi and others, and Dan Geschwind in California. I just want to draw it really to a close now by thanking again Amar Al-Chalabi, Chris Shaw, who was going to come, but his daughter fell off his bike and had to be tended, Bob Brown at Harvard, now at Massachusetts University, a wonderful man who was one of the leaders of the group finding the SOD mutations back in 1993. And Peter Anderson again, and he's sort of Viking, so there you are. He's always traveling. This is the SOD molecule, 166 mutations, and this is to plug for anybody, because time is pressing, everything is there. We set up back in the 1990s with Alex Radunovich a website, and it's now taken over by Amar and others. It's now really a really wonderful website, which I recommend if you have an interest in this condition. And if you really want up-to-date genetics, don't come to me, go to the AllSod database. But while I was at King's, Chris Shaw was very successful in finding 2 new genes. It seems that they explain a minority of mutations, but they're very important because they focus the attention on RNA processing. And this is a paper at the same time from Bob Brown. But that was a stunning success of our policy of creating a cohort of MND patients and getting data, clinical data, and genetics at the same time, although it was very largely due to Chris's efforts himself. I take no credit for that. And one of the great things that's happened just over the last few months is this mutation on chromosome 9. Again, Amar and Chris had been involved in finding that. They didn't get the gene, but does it matter? Others did. They did as well. And this is the most common gene now. And it probably accounts for about up to 10% of all MND. And this is a very rapidly evolving story. That's just AllSod, and I'll just pass on from that. And this is the chromosome 9 story. And I think, again, time is pressing. I'd love to dwell on this, but I think I won't. Just to make this point, that familial MND, and as you know, MND-ALS, is only familial in 5 or 10% of cases, obviously familial, that is, that there's a family history that you can trace back. This chromosome 9 mutation accounts for nearly 30%. And for sporadic, in this series, 6%. So we're beginning to get towards understanding the molecular basis of the sporadic as well as the familial disease. This is the sort of schema that I have with ALS MND and the various variants. Again, I'll just make the point that the familial form is in there. This is a lower motor neuron form. It's all part of the same rubric. And now we know that it's intimately connected with frontotemporal degeneration, frontotemporal dementia, because many of those are due to this chromosome 9 mutation, whether or not they also have MND. And they all have TDP43 changes, apart perhaps from SOD and fuzz mutations, but that's it. Just like to thank the Motor Neuron Disease Association, fundamental to our research. That was the opening of the MND Centre in King's in 1995. And you can see, you recognize this lady, and this was Lady Halifax, who was a very active campaigner for MND. What it allowed us to do was to set up a centre which was people-centred, I hope, and we are working on that here. We don't call it a centre, but our ambition is to create the same philosophy. And so you have the coordinator at the centre, most recently Emma Wiley, a wonderful person to work with, and then you have the multidisciplinary team who are hands-on in the clinic with us, and we have to add the palliative care consultant, and then facilities, people you draw in from time to time as you need them, PEG, RIG, that's the gastrostomy, in non-invasive ventilation, and then the wider community and links with those, and then the world links, if you like, with research and so on and so forth. And that's the sort of model. So the incoming inquiries, single point of contact, and the MND Association, if you like, running through there as a theme. And that's the sort of model. Now, it can be a distributed model, and that's what we're working on here. And I want to finish with some personal tributes. Now, it's not just the food they come for, but it does help. So my advice to aspiring researchers and PIs is always have lots of cakes available, because you can be sure they'll turn up to the meetings then. Lokesh Wijesakira, I described some of his work. Biba Stanton, whose husband Jeremy is here. Satomi. This, our clinic nurse, Marianne Ampong, is extraordinarily energetic. Her work went beyond any call of duty on behalf of the patients. She was known throughout the region for always being available, even though we told her she shouldn't be. Emma Wiley here, our coordinator. Carlos, who was my last PhD student from Chile. The brain bank here, so we integrate the brain bank with everything else. I just mentioned the ways forward. Keith Caldecott, we want to work with on DNA repair. Again, I don't want to dwell on that. Majid Hafez Parast, Sarah Newbury. We have a number of directions, including making stem cells, which Majid is already doing. Epigenetics is going to be important. So we're very excited about that. Experimental medicines or systems neurology, that's really what we're trying to develop. Mara for imaging, because we want to pursue these molecular imaging targets. Dora Duca, we're working on some projects with Nick Medford in neuropsychiatry, and this is Cyril, although it doesn't look like that to me, but it says it is, so I'll take his word. And this is really, how do we take all this negative studies? We only have really so. These are not all the trials. These are many of the trials we've done. Some of these are ongoing. One of the things we'll try to address is why are studies negative? Indeed, are they negative? What sort of signals can we look at rather than discarding a negative on the basis of function, for example? Do we have biological markers? And that's going to be important. So this is the sort of strategy really of feeding in whatever's relevant to this particular study, intervention. Here's your putative ALS, PSP, MSA therapy. And then I would say that one of the things we have to do, and we're working on, is to try and stratify the disease in more intelligent ways. I haven't got time to go into that. So at the moment it feels a bit like Sisyphus carrying the rock up to the top of the mountain only to have it drop down again. But hopefully at some stage we'll reach this stage of being liberated from our Promethean tasks. And that, I think, is there are singularly good prospects here at Brighton and Sussex, and I look forward to collaborating with all the colleagues I mentioned. So thank you very much. Just to say again, you have to, this is again to show just how important it is to keep them well rewarded. You'll notice that the professor is drinking water there. Another one, are they insatiable? Apparently, another one still. But we do occasionally get tough, and if they really, need some help sitting down and writing up. This is just some of the strange people we have to meet with. This is Blash, who's over here in his usual gear. He's rather dressed down this evening from Slovenian national costume. And finally, this is how we make them actually get down to work. This is Camilla Blaine, well wrapped up against the cold at King's. No heating. We only turn it on once they've finished a chapter. And that's my family, Irene, my ex-wife, and the children, and finally end up to say thank you to Catherine and Beth for their tolerance, and for yours as well. And I'm sorry it's gone on a bit longer, but thank you very much for staying and listening.